Hi, everyone. My name is Andrea Diaconescu, and I'm very excited to be presenting an application of connectome neuromass modeling in the context of schizophrenia and psychosis. My presentation is entitled Neurocomputational Modeling for Characterizing Neurocognitive Pathophysiology in Clinical High Risk for Psychosis. I wish I was there in person giving this talk and enjoying the conference with you in Seoul. But unfortunately, I couldn't make it. But at least I'm happy that I get to share this research with you virtually. All right. I have no disclosures. So I'd like to begin my presentation first by painting a picture of schizophrenia. It is one of the most debilitating disorders where symptoms alter one's perception of the world. One patient reported, "My senses are sharpened. Sights and sounds possess a keenness I have never experienced before." Whereas another patient reports, "I had to make sense, any sense, out of all of these uncanny coincidences. And I did it by radically changing my conception of reality." As it is evidenced by these two testimonials, schizophrenia is a disorder that progresses in stages, and each stage is marked by an increase in symptom severity. Now, the most common symptoms are psychosis symptoms or positive symptoms, hallucinations, and delusions, and they have to be present for at least 2 months for a diagnosis to take place. Therefore, diagnosis happens late in the disease progression. The most common forms of treatment include antipsychotic dopamine 2, D2 receptor antagonists, so pharmacological intervention, often coupled with cognitive behavioral therapy. However, certain symptoms are quite resistant to treatment, and schizophrenia is also associated with a reduced life expectancy in spite of these treatments, recurring hospitalization, and occupational disability. In fact, individuals rarely return to the prior level of psychosocial functioning. So a lot of effort in the recent years has been placed on trying to understand pathophysiology that is predictive of psychosis onset in individuals at clinical high risk. So what is clinical high risk for psychosis? So two criteria are used to ascertain heightened risk or clinical high risk for psychosis. The first is a change in subjective experience and behavior, pronounced reduction in functioning of an individual, usually a young adult, accompanied with the either a familial history of psychosis or the presence of brief, limited, intermittent psychotic symptoms or blips, or attenuated psychosis symptoms that do not meet the threshold for diagnosis of schizophrenia. Now, 15 to 18% of those clinical high risk transition to psychosis within 6 months, and then 30% transition within the first 2 years after clinical high risk ascertainment. But even though a small proportion actually transition, the majority continue to experience symptoms and also show a pronounced reduction in psychosocial functioning, even 1 year or 2 years later. So the effort has been placed on trying to identify the underlying pathophysiological mechanisms that predict a reduction in functioning at a later time point in order to better stratify clinical high-risk patients and prioritize treatment. So in my presentation today is basically divided into 4 parts. I'll start off with the theory and the hypothesis of the pathophysiology at different stages in early psychosis. I'll talk about some promising ERP biomarkers that are predictive of psychosocial functioning, and also describe the data set that we have used in our modeling efforts. I will talk about the methods, the computational approach that we took, how we analyze the data, and then discuss the results and the next steps of this project. So the hypothesis that relates to neurobiology is based on this work that is synthesized very well in this publication by Korostil et al. In Biological Psychiatry in 2017, that psychosis is truly a disorder that progresses in stages that are marked by different neurotransmitter and neuromodulatory dysfunction. And the first that sort of starts these cascade of synaptic and neuromodulatory deficits is a glutamatergic synaptic dysfunction that causes a glutamate signaling deficit. And that this triggers a allostatic adaptation, a GABAergic deficit, that and its consequence is increased disinhibition, excitatory inhibitory imbalance that has been demonstrated by looking at oscillations, by looking at brain connectivity. Due to this disinhibition and EI imbalance, the next stage is allostatic adaptation, synaptic downscaling, and programmed synaptic elimination. The consequence of that is the syndrome itself. So we see accelerated brain aging, atrophy, compounding synaptic deficit, tuning deficits that persist, and network functions that decline. Now, if we zoom in on the neurobiology, looking at cortical column, we can try to understand this glutamatergic dysfunction in terms of specific connections between inhibitory interneuron populations, pyramidal cell populations, and spiny stellate cell populations, mediated through AMPA and NMDA receptors that are excitatory, and GABAergic receptors, inhibitory. So the hypothesis is that the excitatory input from pyramidal cell populations onto inhibitory populations is reduced and disrupted in the predrome, in the psychosis predrome. And that leads to a GABAergic dysfunction that essentially causes the disinhibition or reduced inhibitory to pyramidal cell connectivity. And if we look specifically at the interactions between pyramidal to inhibitory interneurons, the idea is that we have these two co-occurring mechanisms: reduced pyramidal to inhibitory interneuron excitation and reduced inhibition from inhibitory interneurons onto pyramidal cells. And this has been proposed as a mechanism to explain disruptions in sensory learning that can be assessed using the auditory mismatch negativity. So the auditory mismatch negativity, examined with EEG, looks at a brain signal of surprise that occurs during probabilistic deviations in the auditory domain in the features of the auditory stimulus, such as frequency or duration. So if we link those two theories of disruption in PI, P2I, pyramidal to inhibitory interactions, the glutamatergic deficits, the hypothesis is that these two mechanisms are occurring at different stages of psychosis progression. So that the first, in the prodrome, we see a glutamatergic dysfunction, reduced excitation from pyramidal cell populations onto inhibitory interneurons. And at the second stage, that explains the disinhibitory effect, excitatory inhibitory imbalance, we have a disinhibition from the inhibitory interneuron cell populations onto the pyramidal cells. So if we translate this into the mesoscale, a simpler model like Janssen and Ritt model, we hypothesize that at the early stages of psychosis in the prodrome, that we should see reduced synaptic gain, reduced P2I connectivity, and then in later stages of the disease progression, in early psychosis or first episode psychosis, we see a disinhibition, in other words, reduced I to P connectivity. And what does that mean in terms of computations and learning processes? And this is where we want to tie the neurobiology to these psychosis symptoms, attenuated psychosis symptoms that have been detected in individuals at clinical high risk for psychosis. So for that, we propose a Bayesian formalism that essentially says that the function of the brain is to make inferences about states in the world and update those predictions through prediction errors that signal the discrepancy between what was hypothesized, what was predicted, and the sensory input. And so at the heart of this, of course, is the Bayes rule. And if we think about it in terms of a hierarchy, then belief updates are hierarchically coupled and change as a function of not only the prediction error, but precisions, two different types of precisions. The sensory precision, which augments the impact that prediction errors have on one's model of the world, and then the denominator, the belief or prediction precision that reduces the impact that prediction errors have. So the belief update, or how one adjusts their model of the world, is dependent not only on the prediction error, but also on this ratio of precision. In other words, if the precision of our prediction is reduced, then we should take incoming prediction errors more into account. And we've used this hierarchical Bayesian theory to try to map learning at different stages of the psychosis progression. So in the prodrome, my senses are sharpened, sights and sounds possess a keenness I haven't experienced before. The hypothesis is that the sensory precision of prediction errors is increased. But we have evidence, in fact, that this is due to a reduction in the precision of the predictions. The reduced precision of predictions, reduced precision of the predictions increases the weighting of incoming prediction errors. In other words, we expect to see an imbalance between the sensory precision and the belief precision that leads to this increased prediction error learning. And we've used this model to understand disruptions in auditory mismatch negativity in the clinical high risk for psychosis populations. What about other ERP biomarkers that might be useful for understanding functional outcomes in clinical high risk? This is what I will talk about next. So one other event-related potential that we have looked at with Michael Kiang at Kiang M is the N400 component. His group has shown that the N400 is a useful biomarker of functional outcomes in biomarker to predict functional outcomes in clinical high-risk populations. But what is the N400 potential? So the N400 ERP occurs in response to any semantic, meaningful stimulus, such as written or spoken words. And it's essentially, you can think of it as a semantic prediction error, or a prediction error that is reflected, that is occurring due to a semantic expectation. So if you're presented with the word whipped cream taste, sweet would not induce a high prediction error, but whipped cream taste anxious, that presentation is unrelated or has lower probability of being associated with whipped cream, and therefore it will induce a prediction error response or a larger N400 amplitude. So we see this larger N400 amplitude for semantically incongruent words. And we call it a semantic prediction error response because it's very similar in that sense, and it's specific to semantic words. So for example, if you create word pairs or use words that don't exist in the English language or the language that the participant can speak, so it's nonsensical gibberish words, you wouldn't see this prediction error response. So it's not just having something that is different or unexpected, but something that is semantically unexpected. Okay. Now, how does it differ in schizophrenia? Well, what you see is that in schizophrenia, patients show larger amplitudes to both strongly and indirectly related targets. So they show a reduced inhibition for unrelated targets. The amplitude deflection is flatter in response to the unrelated targets, whereas controls show that typical N400 response. Now, Jenny Lepock, who's a postdoc with Michael Kiang at Kiang M, looked how much this depends on the type of design one uses. So she also manipulated the stimulus onset asynchrony. So in a paradigm where you have, instead of having to complete sentences, you use word pairs like table, cloth, table, spider, for example, you can manipulate this stimulus onset asynchrony in such a way that if you use a shorter SOA, you should induce sort of more rapid or automatic associations, and at longer SOAs that allow the individual to make semantic expectations of the more likely combination, you should see larger prediction error responses. And this is exactly what they found. So at short SOAs, the authors did not see any group differences in the N400 amplitude. And this N400 response in the short SOA condition actually correlated with the involuntary pre-attentive auditory mismatch negativity component as well. However, at longer SOAs, when individuals are given more time to think about potential association of the word pairs, they saw a larger N400 component. But this N400 component was more attenuated in the clinical high risk for psychosis individuals compared to controls. And the group went one step further and looked at how this baseline N400 response correlated with psychosocial functioning 1 year later. So they had these baseline recordings and they re-invited a group of those at clinical high risk and their overall role and social functioning was assessed. And they looked at whether the degree of N400 attenuation correlated with the psychosocial functioning 1 year later, and they did find a positive correlation. The bigger the priming, the bigger the difference between related and unrelated, the bigger the N400 amplitude, the better the psychosocial functioning 1 year later. And even if you perform a median split and bin the data in terms of low and high social functioning, clinical high risk, you see that those that showed higher social functioning 1 year later showed a bigger N400 response. So this is early evidence that potentially the N400 response could be used as a biomarker of functioning. But what is driving that N400 response? What is the underlying neural mechanisms? We want to understand that, and then we want to address the question, is neurocomputational modeling useful in predicting change in psychosocial functioning 1 year later? So mapping or linking this semantic priming research with our initial hypothesis about the computation that explain disruptions in learning or atypical learning in the psychosis prodrome. So we hypothesized that in psychosis prodrome, we see a disruption in glutamatergic functioning or pyramidal excitation of inhibitory interneurons. And behaviorally, in terms of sensory learning, this is expressed as reduced belief precision that leads to an increase in prediction error learning. And the way that we might express this in terms of the semantic priming effect, the hypothesis is that the top-down backward connectivity is reduced in the clinical high risk, or that would be the neurobiological equivalent of reduced precision of top-down predictions. Reduced connectivity, backward connectivity from prefrontal regions like the dorsolateral prefrontal cortex, parietal, onto visual cortex, visual regions, since this is the response to a visual presentation of the word. So we have semantic areas, prefrontal areas that likely are active, but that we can explain this reduced precision of the prediction through reduced backward connectivity from prefrontal to sensory regions. So the aim of the study is essentially to examine these ERP biomarkers longitudinally. In other words, we record the N400 priming effect at baseline, and we want to use the computational models that are derived from fitting on your computational model to the ERP data to predict functional outcomes 1 year later. The data set that we're using includes 38 matched healthy controls and 42 clinical high risk for psychosis individuals, 27 of them who returned after 1 year for additional clinical assessments. And we use the 4 conditions, related and unrelated pairs with the 2 different stimulus onset asynchrony manipulation. Now, it's important to note that the healthy controls were matched. What this means is that they were matched with respect to cognitive function, verbal and semantic memory to the clinical high-risk population. Okay, so after data preprocessing and artifact rejection that follow the same procedure as in the original study, the model initialization began with initializing values for all the parameters, including neural mass model parameters such as time constants, maximum synaptic potentials, and firing rate parameters, as well as connectivity parameters. And this allowed us to simulate the N400 evoked activity. In order to simulate the neural activity, we used the Janssen and Ritt model at each node of the connectome. So in the Janssen and Ritt model, the response of the postsynaptic membrane potential V of T to an incoming presynaptic input is described by a second-order differential equation, and the simulation involves solving the model's differential equations to predict the neuronal population's response to the input, including the N400 evoked potential. We integrate these nodes into a network based on the connectivity matrix that was derived from prior neuroimaging data. And then we project the neural activity to EEG using a lead field matrix. And this allowed us to generate synthetic ERPs that we can directly compare to the empirical ones. So in the study, we utilized diffusion-weighted MRI data from the Human Connectome project to create a detailed anatomical map of brain structural connectivity. We used the Schizophr Atlas for atlas-based parcellation that divides the brain into 200 regions grouped into 7 functional networks. And this provided us with a standardized way to define the brain nodes and their connections. So each node in the connectome was modeled using the Janssen and Ritt neural mass model that describes the dynamics of local neural populations through differential equations. And it consists of 3 interconnected populations: pyramidal neurons, excitatory interneurons or spinal stellate cells, and inhibitory interneurons. So the Janssen and Ritt represents a single cortical column that encapsulates the collective activity of large populations of neurons. So pyramidal neurons receive inputs from both the excitatory and inhibitory interneurons. And these are the principal output neurons that project to other regions within and outside the cortex. Excitatory interneurons receive input from pyramidal neurons and send excitatory feedback to pyramidal neurons. And for both P and E populations, we also estimate HE, which is the maximum amplitude of the excitatory postsynaptic potentials. Inhibitory interneurons, they receive input from pyramidal neurons and send inhibitory feedback back to pyramidal neurons. And then we also estimate HI, which is the maximum amplitude of the inhibitory postsynaptic potential. The intrinsic connectivity of these populations is governed by parameters C1 to C4. And then we have a sigmoidal transfer function, S of B, where the output of each neural population is modeled using this transfer function that essentially converts the membrane potential V into a firing rate. As a forward model, we use the boundary element method model to compute the electrical potential on the scalp that results from neural activity within the brain. So in other words, as a forward model, we use the lead field matrix to map neuronal activity from the brain to the scalp, to sensor space. Okay, so putting everything together, basically after simulating N400 data, the optimization process starts by defining a loss function that quantifies the difference between the simulated ERPs and the empirical ERPs based on cosine similarity. Then we do parameter adjustments. So we use optimization algorithms such as stochastic gradient descent or ADAM to adjust the model parameters iteratively. And the goal is to minimize the loss function, so improving the match between the simulated and the empirical data. So we also apply rigorization techniques to prevent overfitting, using penalty terms to the loss function to discourage excessive complexity in parameter estimates. So what we're estimating in terms of parameters when fitting this model to the N400 ERP data are the intrinsic connectivity parameters C1 to C4, as well as the decay rate and the amplitude of the excitatory and inhibitory postsynaptic potentials. We perform 30 simulations by varying the initialization values, and we take the mean to assess condition, group effects, and correlations with psychosocial functioning, and we investigated 4 different conditions, as well as global functioning at baseline and 1 year later. So it's important to note that parameters C1 to C4 that capture intrinsic connectivity in the Janssen-Ritt neural mass model describe synaptic coupling strengths within each specific node of the connectome. So they define how different neuronal populations, pyramidal, excitatory interneurons, inhibitory interneurons, interact within a single cortical region. While C1 and C4 are set for each node individually, they typically have the same values across all the nodes in the model. So this uniformity assumes that basic synaptic dynamics are similar across different regions of the cortex. Basically, we estimate C1 parameter that is the pyramidal to excitatory intrinsic connection that captures the effect of the input onto the network. The C2 parameter, excitatory to pyramidal connectivity. So here, when an increase in this E to P or C2 parameter reflects an increase in synaptic gain, making cells more responsive to incoming input. We also estimate C3 that captures pyramidal to inhibitory connectivity or the excitation of inhibitory interneurons. And we hypothesized previously that this is the intrinsic connection to be disrupted in the psychosis prodrome that reflects the glutamatergic dysfunction. C4 parameter captures the connectivity from inhibitory onto pyramidal cells, inhibitory interneurons onto pyramidal cells. So a reduction in I to P connectivity reflects a reduction in synaptic gain, making the cell less responsive to input. And here we hypothesize that the allostatic adaptation is reflected through a reduction in the I to P connectivity, leading to disinhibition. We also estimate the inhibitory synaptic gain, the amplitude, as well as the decay rate, as well as the excitatory gain and decay rate for the excitatory postsynaptic potential. And finally, we use partial least squares to look at both condition differences, group differences, and correlations between model parameters and psychosocial functioning. We employ, of course, both mean-centered and behavioral PLS approaches to identify these differences and relationships. And that brings me to the result, finally ending with the outlook. So first of all, when looking at the event-related potentials across conditions and groups, we could replicate the N400 group and condition differences, where we see a lack of group difference in the short SOA condition and a dampening of the N400 amplitude, a dampening of this priming effect in the clinical high risk for psychosis group compared to the control. Now, when we looked at condition differences in the model parameters, we saw a significant difference between the short SOA and the long SOA conditions. And when we looked at the bootstrap weights of these parameters, where bootstrap values that are larger than 2.5, reflecting reliability across our participants, we see that parameters C4, B, and B that capture the amplitude and the decay rate of the IPSP tend to be the parameters that maximally differentiate those two conditions across all participants. What they reflect is that we see an increased inhibition through higher C4 and B parameters in the short compared to the long SOA conditions. So reduced inhibition in the long SOA condition reflects larger semantic associations and representations in the larger SOA condition. How about group differences? So we observed group differences specifically for the unrelated long condition, where we observe this semantic prediction error response or the N400 priming effect. We see differences between clinical high risk and controls in the parameters that capture the synaptic gains, the inhibitory and excitatory. And these are the bootstrap weights of the parameters. So again, we're looking at values that exceed 2.5. And what this tells us here is that in the clinical high risk for psychosis compared to the control population, we see a reduction in synaptic gain. So faster decay rate of EPSPs and larger inhibition, larger amplitude of the IPSPs for clinical high risk compared to controls. And the interpretation is that reduced synaptic gain reflects this reduced semantic prediction error response that we see in the clinical high risk population. We also see a group difference in the backwards in degree connectivity between several brain regions Again, we can look using the bootstrap weights, we can look at what brain regions are driving these differences in backward connectivity. And specifically drawing your attention into the regions in blue that show a reduced backward connectivity in the clinical high risk for psychosis group compared to healthy controls. We see reduced backward connectivity from the medial prefrontal, dorsolateral PFC, and supramarginal gyrus to visual regions. In terms of the location of these connectivity differences, seems to be quite in line with the semantic priming task that participants are performing. So just to remind you, originally we hypothesized that this reduced precision in top-down predictions explains or computationally explains this dampening in the semantic prediction error response. And this can be expressed through reduction in top-down modulation from the VLPFC to the posterior parietal cortex to the visual cortex. And that explains this reduced prediction error response. Now, what about 1 year later? When we looked at the participants, the subgroup that we were able to contact and assess after 1 year, we have a substantially smaller population that we can examine, but we see similar condition differences in the smaller clinical high-risk group compared to those that were assessed at baseline. So similar condition differences, those two subgroups. Now, what about correlations with psychosocial functioning? In this case, we see two parameters standing out and showing correlations with a change in psychosocial functioning 1 year later. And specifically, we're looking at the difference between the unrelated and related long condition. We want to explain this dampening of the N400 response. And what we see is that there's a reduction in the P to I connectivity and an increase in the E to P excitation in response to the semantic prediction error responses associated with poor functioning 1 year later. So suggesting that this disruption in glutamatergic functioning, reduced excitation of pyramidal cells to inhibitory interneurons, plus increased excitation through the excitatory interneurons onto pyramidal cells is associated with poor functioning 1 year later. So what our research suggests thus far is that we see we have evidence for a glutamatergic deficit in the CIHR, clinical high risk for psychosis population, that is associated with psychosocial functioning, a reduced top-down modulation that explains the group difference between patients and clinical high risk for psychosis individuals and controls, that explains this reduction in the N400 priming effect. What are the implications of this? So a hypofunctioning, what this might suggest is the hypofunctioning of NMDA receptors or inhibitory interneurons. And this impaired activation of inhibitory interneurons may capture increased excitatory inhibitory imbalance. With this modeling framework, we cannot really examine or address the hypothesis regarding NMDA receptor hypofunction, but we are planning to assess this using conductance-based model in the Connectome whole brain framework as well. And the outlook, the future direction of this work is to integrate this approach, so looking at ERP and computational biomarkers with speech coherence parameters, and relate the semantic priming deficits with specific speech markers that are linked to thought disorder and positive and negative symptoms in early psychosis, and test whether these parameters can improve our ability to predict psychosocial functioning 1 year later. I want to end the presentation by thanking my team, the Cognitive Network Modeling Group at CAMH, as well as my collaborators, in particular, John Griffiths and Michael Kiang, with whom worked on this specific semantic priming EEG effect. And I would like to also acknowledge my collaborators in Toronto, at Unity Health and University Health Network, as well as those in Europe. And I'd like to also acknowledge my funding sources. And thank you again for your attention.